Welcome to the Deep Dive. Today, we're diving into the latest ophthalmology research with the insights of Professor Dr. Suhail Sarwar.
Right.
Now, this deep dive is really for ophthalmologists and other healthcare professionals. We're going to be using the precise language of our field. If you're looking for something more patient-friendly,
Right.
Professor Sarwar has a great playlist on eye diseases for the general public.
Yeah.
But we're going to dissect the latest research from international journals.
Exactly. Think of this as like your accelerated update on what's new and important straight from the source.
Okay, let's start with this study on visual field mapping in XLRP. They're working on generating hills of vision. It sounds really cool. Imagine being able to visualize a patient's remaining vision as like a 3D landscape.
Yeah, it's a really powerful concept, and this study uses data from 78 eyes of XLRP patients.
Yeah.
And it digs into the accuracy of different mathematical methods for creating these 3D representations. They tested 3 techniques: thin plate spline, or TPS, Gaussian, and universal Kriging interpolation. Think of these as like sophisticated algorithms that fill in the gaps from a standard visual field test, creating a smooth, continuous visual field surface.
So it's more than just like connecting the dots. It's building a detailed topographical map of their vision. And did they actually calculate like the volume of these hills?
Yes, they did. They calculated the volume for the entire visual field and also for a specific 30-degree region.
Right.
And they used this statistical method called Bland-Altman analysis to assess the agreement between the different measurement techniques.
Okay.
What they found was that while all 3 methods showed good correlation, TPS had tighter limits of agreement compared to Gaussian and universal Kriging.
So TPS was more accurate.
Yes, indicating greater accuracy.
That's a really critical detail for us as clinicians, right? Because knowing that the TPS method is more reliable means that we can have more confidence in these hills of vision it generates. This could lead to like more precise and personalized rehabilitation strategies for XLRP patients.
Absolutely. Imagine being able to tailor visual aids or rehabilitation exercises based on the exact contours of their remaining vision. That level of precision could be a game changer.
This is so exciting. Okay, let's shift gears and talk about a breakthrough in optogenetic therapy. This study investigated a novel therapy called MCO010 in 2 mouse models of retinitis pigmentosa at RD1 and RD10. Now, I know these models are essential for preclinical research, but can you remind us why they are so crucial in mimicking human RT progression?
Of course. These mouse models are invaluable because they closely mirror the degenerative processes and genetic underpinnings of human RP. This makes them a critical testing ground before transitioning to human trials.
Makes sense.
In this study, MCO010 was delivered via intravitreal injection, and remarkably, around 80% of bipolar cells were successfully transduced.
Okay, so that's significant coverage within the target cell population. What were the functional outcomes of this treatment?
The results are quite remarkable. The study demonstrated significant improvements in visual function. For example, the mice showed increased head movements in response to visual stimuli. Think of it as regaining the ability to detect and react to objects in their environment.
Okay.
They also performed significantly better in a water maze test, navigating the maze much faster, which points towards improved spatial awareness and vision-guided behavior.
Oh, that's fantastic. What was particularly striking to you about these findings?
Well, what's particularly compelling are the electrophysiological findings. The researchers went beyond behavioral observations to examine the electrical activity in both the retina and the visual cortex. They recorded visually evoked potentials, or VEPs, which essentially showed the electrical responses of the brain to visual stimuli. And in these RD1 mice, which typically have minimal, if any, visual responses due to severe retinal degeneration, they saw clear restored VEP signals after treatment.
Wow, so we're potentially seeing the rewiring of the visual pathway, bypassing those damaged photoreceptors and directly stimulating the remaining healthy cells.
Yes, exactly.
And this is further supported by the histological evidence they provided. Those images showing expression of the MCO010 gene in bipolar cells confirm that the therapy reached its intended target.
Precisely. And the fact that they achieved transduction in about 80% of bipolar cells underscores the efficiency of their delivery method and the viral vector used.
That's great.
This combination of robust transduction, restored visual function, and a favorable safety profile makes this a development to watch closely. And remember, they observed no toxicity or adverse effects even when exposed to intense light conditions.
That's incredible. This is definitely a game changer. Now for our next deep dive, let's explore a neuroprotective strategy, focusing on the TRKC.T1 receptor.
Okay. This is a fascinating approach because it targets a specific molecular pathway implicated in photoreceptor death. It involves inhibiting a receptor called TRKC.T1, which is upregulated in Muller glia, in RP. Its activation by a molecule called NT3 actually contributes to the death of photoreceptor cells.
So essentially, we're trying to block this harmful pathway that's driving the degeneration of those precious photoreceptors. What did this study reveal about this approach?
Well, the researchers developed small molecule antagonists that specifically target and block TRKC-C2 without interfering with other related receptors that are important for normal neuronal function.
Got it.
They tested these antagonists in the RHOP347S mouse model of RP, a model known for its aggressive form of retinal degeneration.
Okay. And what were the outcomes of using these antagonists? Were they able to protect the photoreceptors?
The results were highly encouraging. They found a significant reduction in photoreceptor death in the treated eyes. And it wasn't just functional data, they had histological evidence to support their findings.
Okay.
The study included images showcasing a thicker layer of surviving photoreceptors in the treated eyes compared to the control group. It's a powerful visual confirmation of the neuroprotective effect.
That's incredible. It sounds like we are moving towards a more targeted and precise approach to neuroprotection in RP. Now let's move on to a study that delves into the complex world of protein misfolding, a key player in many retinal diseases.
Right.
This study explores the potential of archaeal enzymes, specifically a complex called PNET, to address this issue.
This study is particularly intriguing because it borrows tools from extremophiles, organisms that thrive in some of the harshest environments on Earth, and applies them to combat human disease.
Wow.
These archaeal enzymes have evolved to function under extreme conditions of temperature and pressure, making them incredibly robust and resilient.
Okay.
The idea is to leverage this resilience to target and clear the stubborn protein aggregates that contribute to retinal degeneration.
Sounds like we're bringing in a specialized cleanup crew, equipped to handle the toughest challenges. What makes PNET, this archaeal unfolded protease complex, particularly well-suited for this task?
Well, PNET possesses this dual capability of both unfolding misfolded proteins, giving them a chance to refold correctly, and degrading those that are beyond repair.
Okay, interesting.
They tested PNET in the P23H rhodopsin mouse model of RP, known for its particularly aggressive form of retinal degeneration.
What did they observe when they used PNET in this model?
They found that PNET increased the viability of rod photoreceptors. This suggests that it was effective in recognizing, targeting, and clearing those misfolded rhodopsin proteins, potentially slowing the degenerative cascade.
Wow, it's like opening up a whole new avenue of therapeutic possibilities. If we can successfully harness these archaeal enzymes, we could potentially target a wide range of retinal diseases, where protein misfolding is a key factor. Now, before we wrap up this part of our deep dive, I want to highlight one more groundbreaking study that used CRISPR base editing to correct the E150K rhodopsin mutation. This is a perfect example of the rapid advancements being made in gene editing technology.
Absolutely. The study employed adenine base editing, a technique that allows for precise A to G conversions in the DNA sequence.
Wow.
And they specifically targeted the E150K mutation, a known cause of autosomal recessive RP.
So they literally went in and fixed the typo in the genetic code.
Yeah.
And they accomplished this directly in the eyes of mice carrying the E150K mutation using dual adeno-associated viruses, or AAVs, to deliver the base editing machinery to the retinal cells. What were the outcomes of this approach? Did they achieve correction of the mutation?
The results were quite impressive. They achieved significant levels of correction, up to 44% in some cases, and even more importantly, they observed partial restoration of both retinal function and structure.
Wow.
Electroretinography, or ERG, showed improvements in the retinal responses, indicating enhanced signaling within the retina, and optical coherence tomography, or OCT imaging, revealed preservation of the retinal layers.
That's remarkable. We're talking about not just correcting the genetic defect, but also seeing functional and structural recovery. This really highlights the potential of base editing as a powerful tool for treating inherited retinal diseases.
It's a glimpse into the future of personalized medicine, where we can tailor therapies to an individual's specific genetic makeup.
This has been an incredible overview of some truly groundbreaking research. Now let's take a closer look at some of the nuances and implications of these studies in the next part of our deep dive.
So let's delve a bit deeper into some of these studies and really unpack the details for our fellow experts. Starting with that visual field mapping study, Their meticulous approach to comparing those interpolation methods was really impressive. Remember, they used Bland-Altman
Yeah,
analysis?
yeah, that's always a good sign in my book. They're essentially looking for any systematic biases between the methods to see if one consistently overestimated or underestimated the others.
Exactly, and the Bland-Altman plots really help visualize that agreement. And remember how the TPS method had tighter limits of agreement? Well, it turned out to be more accurate by an average deviation of 2 degrees, compared to 4 degrees for the Gaussian and universal Kriging methods.
Wow.
This is significant because it means we can have more confidence in those hills of vision generated using TPS, which translates to more precise and personalized rehabilitation strategies for our XLRP patients.
Yeah, that level of accuracy could really transform how we approach rehabilitation.
Yeah.
Now, what about that optogenetic therapy study? What really stood out to you?
The meticulous detail they employed to characterize the effects of MCO010 was truly impressive. They didn't stop at just behavioral observations. They went deep into the electrophysiology of both the retina and the visual cortex.
Yeah, those visually evoked potential recordings were really compelling. Seeing those clear electrical responses in the visual cortex post-treatment is a strong indication of restored visual processing.
Absolutely. And remember, this was in Ordo 1 mice, which typically have extremely limited, if any, visual responses. So to see these restored VEP signals is remarkable.
It's like they're rewiring the visual pathway, bypassing those damaged photoreceptors and directly stimulating the remaining healthy cells. And they didn't stop there. The histological evidence they provided, those images showcasing expression of the MCO010 gene in bipolar cells, visually confirms that the therapy reached its intended target.
Precisely. And remember, they achieved transduction in approximately 80% of the bipolar cells. That's a high level of efficiency, which speaks volumes about the effectiveness of their delivery method and the viral vector they used. It's like a beautiful synergy of targeted delivery, a robust transduction, restored function, and a favorable safety profile.
It's like watching all the pieces fall perfectly into place. Now, turning our attention to the neuroprotective study with the TRKC.T1 antagonists. What are your thoughts on this approach?
It's a brilliant strategy. They're targeting a specific pathway known to be involved in photoreceptor death, and the beauty of it lies in its selectivity. These antagonists specifically block TRKC.T1 without interfering with other TRK receptors, which are crucial for other neuronal functions.
It's like a precision strike against a specific target while minimizing collateral damage. And they didn't rely solely on functional assays to demonstrate the protective effect. They provided histological evidence of preserved photoreceptors.
Exactly. Those images showing thicker outer nuclear layers in the treated eyes provide compelling visual proof that these antagonists are working.
This potent combination of molecular targeting and histological validation is incredibly promising. Now let's shift gears and explore the fascinating world of arcyl enzymes. What are your initial impressions of this innovative approach with PANET?
It's a bold move. They're borrowing tools from extremophiles, organisms that thrive in extreme environments, and applying them to human disease. These archaeal enzymes have evolved to function under intense stress, including extreme temperatures and pressures. The idea is to harness their robustness and resilience to tackle those stubborn protein aggregates that accumulate in retinal diseases.
It's like sending in a specialized cleanup crew to deal with a hazardous material spill. They're built for the toughest challenges. Now, just for clarification, can you remind us what an unfoldase and a protease are?
Sure. An unfoldase, as the name suggests, is an enzyme that assists in unfolding misfolded proteins, giving them a chance to refold correctly. On the other hand, a protease is an enzyme that breaks down proteins into smaller fragments. So PANET, with its dual capabilities, can both unfold and degrade misfolded proteins.
A two-pronged attack. And they chose to test PANET in the P23H rhodopsin mouse model of RP, known for its aggressive form of retinal degeneration. What were the outcomes?
They discovered that PANET increased the viability of rod photoreceptors, suggesting that it was effective in targeting and clearing those misfolded rhodopsin proteins, potentially mitigating the progression of the disease.
This opens up a whole new realm of therapeutic possibilities. Now let's shift our focus back to that incredible study that utilized CRISPR-based editing to correct the E150K rhodopsin mutation. It's a perfect example of the rapid advancements in gene editing technology.
Absolutely. This study employed a specific type of base editing known as adenine base editing, which enables precise A to G conversions in the DNA sequence. They specifically targeted the E150K mutation, which is a known cause of autosomal recessive RP.
So they went in and corrected the typo in the genetic code, and they accomplished this directly in the eyes of mice carrying the E150K mutation, using dual adeno-associated viruses, or AAVs, to deliver the base editing machinery to the retinal cells. What were the outcomes of this in vivo approach?
They achieved a remarkable level of correction, up to 44% in some cases. And importantly, they demonstrated partial restoration of both retinal function and structure. They observed improvements in the electroretinography, or ERG, responses, indicating improved retinal signaling, and they also noted preservation of retinal layers on optical coherence tomography, or OCT, imaging.
It's like they took a precise scalpel to the genetic code and repaired the damaged section. This is a powerful demonstration of the potential of base editing to effectively target and correct disease-causing mutations in the retina.
Absolutely. It's still early-stage research, but the implications are enormous. Imagine the possibilities if we can refine this technology and translate it to human patients.
It's a glimpse into the future of personalized medicine. Now let's turn our attention to another group of studies that explore the diagnostic and prognostic aspects of RP. The first one delves into the use of microperimetry, a technique that assesses visual function at a very detailed level.
Microperimetry goes beyond standard visual field testing, providing a more nuanced map of retinal sensitivity. This particular study explored its use in RP patients, looking for correlations between microperimetry findings and structural damage observed on OCT and autofluorescence imaging.
So they're trying to connect the dots between functional and structural abnormalities. What were their key findings?
They found significant correlations, suggesting that microperimetry can offer valuable insights into the extent and progression of retinal damage in RP. For instance, they noticed that areas of reduced retinal sensitivity on microperimetry often corresponded to areas of thinning or disruption of retinal layers on OCT.
It's like having a more sensitive lens to detect those early signs of retinal damage, potentially enabling earlier intervention.
Precisely. The study also emphasized the potential of microperimetry to monitor disease progression over time, offering a more sensitive measure of functional decline compared to standard visual acuity testing.
This is vital information for both clinicians and researchers. Now let's move on to a study that investigated the efficacy and safety of minocycline in RP treatment. Minocycline has been on the radar for its potential neuroprotective properties.
Indeed, this study explored its specific effects in RP patients. Minocycline belongs to a class of drugs called tetracyclines, known for their anti-inflammatory and antimicrobial properties. However, minocycline in particular has shown promise in safeguarding neurons from damage.
So the concept is to harness its neuroprotective effects to slow down or prevent the progression of retinal degeneration. What did this study reveal about its potential benefits and risks?
While still in its early stages, this study produced some promising preliminary data. They observed some improvements in visual function and some evidence of reduced retinal damage in patients treated with minocycline.
That's encouraging, but we always have to consider safety. Tetracyclines as a class can have side effects.
True, but in this particular study, minocycline was generally well tolerated, with no significant adverse events reported. However, it's crucial to emphasize that more research is needed to fully evaluate both the efficacy and the long-term safety of minocycline in RP treatment.
It's a promising lead, but as with any new therapy, caution is warranted. Now let's broaden our perspective and consider a comprehensive literature review that highlighted current non-surgical approaches for managing RP.
This review provided a valuable overview of the current therapeutic landscape, encompassing a range of approaches, including gene therapy, stem cell therapy, and neuroprotective agents. It emphasized the significance of a multi-pronged approach, targeting various facets of the disease process. It underscores the fact that there's no one-size-fits-all solution for RP. Each patient's case is unique, and the optimal treatment strategy might involve a combination of therapies.
Absolutely. This review emphasized the rapid pace of research in this field, highlighting new discoveries and advancements emerging constantly. It's a testament to the commitment and ingenuity of scientists worldwide, tirelessly seeking new ways to combat these devastating diseases.
It's truly an inspiring time to be in ophthalmology. We're witnessing a convergence of scientific disciplines, from genetics and molecular biology to bioengineering and nanotechnology, all focused on developing innovative treatments for retinal diseases.
It truly is. And this brings us to a crucial aspect, the need for better diagnostic tools and prognostic markers. Early and accurate diagnosis is paramount for timely intervention and informed treatment decisions. Studies like the one we discussed on microperimetry are shedding light on potential new avenues in this area. It feels like we are moving from a blurry snapshot to a high-definition image of the retina. This particular study also delved into a specific aspect of microperimetry, the analysis of deep scotoma points. Can you elaborate on what these are and why they are so important?
Sure. Deep scotoma points are areas within the visual field where sensitivity to light is severely diminished or completely absent. Essentially, they represent areas of significant retinal damage. This study discovered that the number of deep scotoma points, particularly those located in the parafoveal region, the critical area surrounding the fovea responsible for our central vision, was strongly correlated with visual acuity.
So it's a more precise indicator of the actual vision loss a patient is experiencing, going beyond simply measuring overall retinal sensitivity.
Exactly. It provides a more refined measure of functional impairment, and this study also linked deep scotoma counts with autofluorescence patterns, revealing a connection between functional deficits and structural alterations in the retina.
It's fascinating how these studies are helping us connect the dots between a patient's subjective visual experience and the objective findings from imaging studies. It's all about building a more comprehensive understanding of the disease process. Now let's circle back to those exciting new treatment avenues. We've discussed gene therapy, neuroprotection, protein misfolding, and base editing, all incredibly promising approaches. But what about stem cell therapy? It holds immense potential for treating retinal diseases, doesn't it?
It certainly does. Well, stem cell therapy for RP is still in its early stages. There have been some encouraging preclinical and clinical studies. The fundamental idea is to leverage stem cells, those remarkable cells with the ability to differentiate into various cell types, to either replace damaged retinal cells or provide support to the remaining healthy cells.
It's like replenishing lost troops or sending reinforcements to the ones still fighting. What types of stem cells are currently being explored for this purpose?
Researchers are investigating various types of stem cells, including embryonic stem cells and induced pluripotent stem cells, or iPSCs, and mesenchymal stem cells, or MSCs. Each type has its own unique advantages and challenges. And scientists are actively working to determine the optimal cell type and delivery method for treating RP.
And there are clinical trials underway testing the safety and efficacy of stem cell therapy in human patients.
Yes, there are. And while it's still too early to draw definitive conclusions, the preliminary results are promising. Some studies have reported improvements in visual function and some evidence of structural preservation in patients treated with stem cells.
It's a glimmer of hope for those who have been told there's no cure for RP. It's a testament to the relentless pursuit of new possibilities in this field. Speaking of pushing boundaries, have you come across any studies that combine stem cell therapy with gene editing?
As a matter of fact, I have. One study took MSCs, a type of adult stem cell that is readily accessible, and genetically modified them to express a therapeutic protein. They then transplanted these modified MSCs into the eyes of mice with RPA.
That's incredible. They essentially created tiny factories within the eye to produce those therapeutic proteins right where they're needed. Did they see any positive effects from this approach?
They did. They selected a protein known to promote photoreceptor survival and function, and the results were impressive. They observed significant improvements in both retinal structure and function in the treated mice.
This is a powerful demonstration of the synergistic potential of combining cutting-edge technologies. It feels like we are entering a new era of therapeutic possibilities where we can not only replace damaged cells, but also enhance their function and resilience.
It is indeed a thrilling time to be a part of this field, and I am confident that we will continue to witness groundbreaking discoveries and advancements in the years to come.
As we wrap up this deep dive into the latest ophthalmology research, I'm left with a sense of awe and optimism. The pace of discovery is truly remarkable, and the future holds immense promise for our patients. Remember to stay curious, stay engaged, and keep pushing the boundaries of what's possible in our field. Until next time.